Alright, so now that we've got kind of the basics about the nervous tissue and neural divisions and whatnot, now let's dive deeper into the anatomy of nervous tissue. So this is where we're going to get into the different types of neurons. We'll talk about the basics of a neuron, which we've kind of covered a little bit, but we're going to get into the different types of neurons in terms of structure and function. And then we're going to get into all the different glial cells, both in the central and the peripheral nervous systems, and then talk about myelination and axon regeneration. So just as a reminder, in case you forgot, right, neurons are responsible for sending electrical signals to communicate. So what we call an action potential, right? So it's like an electrochemical signal, right, electrochemical current that is transmitted throughout the neuron to communicate with something else. And so this is what's going to be used to communicate about sensations. It's what we use to produce movement in response to that, to cause glands, body parts to do something, right? It also is like part of your thought process, and you're using these guys to, make sense of what you're hearing right now, right? These cells are massive in comparison to other cells. If you remember looking at this tissue, the spinal smear in lab, you may have noticed that the neurons are massive in comparison to the glial cells. And they're non-mitotic, meaning these cells do not go through mitosis the way other cells do. That's new, that's different, right? So it's they're a little bit strange, they're a little bit different compared to a lot of the cells we've talked about in the generic cell you've learned about in the past. Now the glial cells, sometimes called glia, sometimes called neuroglia, glial cells, you can use those terms interchangeably. I don't care. I might even use them interchangeably. More than likely, I'll use neuroglia or glia just to keep it consistent if possible, but you could see any of them. These are all supporting cells, so they're going to do various jobs to help support the neurons to ensure the neurons can keep doing their jobs. So this is that picture, right? This is similar to what we saw in lab, a picture of that neuron. You can see it's huge right there in the middle. And all of those little purple circles are the glia. So a huge difference in terms of their size and shape. So some of this is going to be review from our last video and from when we talked about nervous tissue way back towards the beginning of the semester, but the parts of the neuron, we've talked about the soma, which is the cell body where we find the nucleus in that neuron. The dendrites are the extensions that are going to receive information from other, other parts, other neurons, receptors, something, right? And then the axon is going to be the extension that carries information away. A neuron can have multiple dendrites, but neurons generally only have one axon, right? There is a point where the axon is going to be coming from that soma. That's called the axon hillock. And that's going to be covered with glial cells that actually make the myelin. So the myelin is coming from glial cells. All the way down the end, we get to the axon terminal. We have all those little branches at the end of the axon terminal where we can communicate with other neurons or muscles or glands or whatever we're talking to. And you'll notice that at the very end of those axon terminal buds, like the little branches at the end, they kind of bulb out, and that's because they are a synaptic end bulb. And then if you may have noticed when we were looking at the axon earlier and I was pointing out the myelin sheath, that myelin sheath was not continuous, that there were gaps in between, right? There were breaks in that myelin sheath. Each of those breaks or gaps is called the nodes of Ranvier, right? So those nodes of Ranvier are the gaps between them. We're going to talk about, at some point, we'll probably talk about why we have those nodes. We're not going to worry about it just right now. Right, so here is our nice little pretty picture of a standard generic neuron, right? So our cell body right here, all these dendrites coming off of it. Here's the axon hillock, right where the soma and the axon kind of connect. The axon itself coming all the way down, the axon terminal, and then the little bulges at the end, the synaptic end bulbs. And like I pointed out earlier, the last slide, the myelin sheath is not continuous. We have those gaps, and those gaps are called the nodes of Ranvier. And in this case, we're seeing one of our glial cells called an oligodendrocyte. Right, that's one of the specific types of glial cells that can produce that myelin sheath. So now let's go ahead and talk about the different types of neurons. So at this point, we've been looking at the generic neuron, right? Nothing special, just the same basic picture over and over again. But there's actually more than one type. And so we have these three main categories of neurons: unipolar, bipolar, and multipolar. And this has to do with how many processes we have. So in the case of the unipolar neuron, we have one process. That means the dendrites and the axons are combined together, coming from the soma. This is what we see when we look at ganglia, right? So we see that the cell body is in the ganglion, and then the process, the axon dendrites, are coming from that, extending out. Bipolar, we have the axon, we have the dendrite, right? So there's a cell body, we have one process that's an axon, we also have a dendrite. So we see these in like the retina, we see these in like the olfactory epithelium for like your sense of smell. And then lastly, we have the multipolar neurons. This is where we have lots of processes from the soma. So you have that axon, but you have like a bunch of like dendrites. These guys we find all over the place, right? We can find these all over the place within the central nervous system. So most cases, multipolar neurons are going to be what you're looking at, but you could be looking at a unipolar or a bipolar at some point. So here is what we're looking at, right? So here's our unipolar. Looks a little weird, right? So we have like the one process, one is a dendrite, one like this side's like an axon, bring information in, process, send it out, but it's kind of along the same thing. Here's the bipolar one, cell body's kind of in the middle, right? So we have like a dendrite, soma, axon, and then this is our generic, you know, what we've been looking at and thinking of as a neuron is multipolar, where we have a bunch of dendrites coming in, and then one single axon with its terminal. So we can also classify neurons based on where we find them, or even like who found them. So they might be named after someone, sometimes after what they do, or even the chemicals they use to communicate with each other. So like what neurotransmitters, like there's all sorts of ways that we can classify neurons. We're not going to get into all the classifications because that's brutal, but you should know the ones that we have gone over so far. That's for sure. So like here's just some examples. I'm not going to get super caught up in these ones just like yet. Make sure that you can identify at least by shape though, for sure. But like here's some of those interesting other ones, right? This pyramidal one that we see in the cerebral cortex, right? So it's a little bit wonky looking, right? We see this Purkinje cell. This is in the cerebellar cortex, right? Very branched, right? Very branched. And then over here, we see the olfactory cells, right, in the olfactory bulbs, right? Bam. We can also classify neurons on function, and this is one type of classification you definitely do need to know. Do not ignore this, because this has to do with how the neuron is functioning in terms of what is its job in the grand scheme of the nervous system. And it really comes down to the three. And you'll notice these names are very similar, right? We talked about sensation, integration, and response. Sensory neurons, interneurons, motor neurons were named after what we're doing in that major function of the nervous system, right? Sensory neurons are doing just that. They're receiving sensory information from the stimulus, right? They're helping you perceive pressure, temperature, touch, pain, right? Sensory neurons can be varied in terms of their shape, unipolar, bipolar, or multipolar. But these are ones that help you bring information in, either about the world around you or within you. The interneurons, they're going to do the integrative functions, the retrieval, the processing, the storage of information. They're also going to help communicate between the sensory and motor neurons. Interneurons, this is a big thing, interneurons are only found in the central nervous system. You will not find interneurons in the peripheral nervous system. That's an easy thing to ask about. You should know that one. We see multipolar neurons for these guys, and most of the neurons we have are interneurons, right? So this is super common. And then the last one are the motor neurons. This is what's going to take information from the brain and spinal cord and communicate that information to our muscles, glands, various body parts. We can find them in the central nervous system, or we can find them in our autonomic ganglia. These guys are multipolar neurons. So really, if you've noticed, the bipolar, the unipolar ones are pretty much sensory neurons only. So if I was asking you, hey, you look under a microscope, you see a unipolar neuron, what type is it most likely to be? You should be able to tell me sensory. Okay, so now we can move on from the neurons to the supportive cells, the glia, the neuroglia, the glial cells, right? So we're going to talk about all the different types that we find in the central nervous system, then we'll talk about some of the ones we find in the peripheral. Let's start with the central nervous system ones first, in which we have 4. We have the astrocytes. The astrocytes, they're going to mainly be helping contribute to maintaining the blood-brain barrier, so making sure that we have a barrier between the rest of the body, basically, and the brain. The oligodendrocytes. The oligodendrocytes, we saw those guys before and that they were producing the myelin sheath, so their job is to insulate the axons. And then the microglia, they're called micro because they're smaller than even the other glial cells, so they're really little. These guys are macrophages that will basically get rid of any cells like pathogens that could cause disease, anything that's not supposed to be hanging out there, right? And then lastly, the ependymal cells, these will help filter out blood. They're going to actually make cerebrospinal fluid, or CSF, which is a fluid that circulates throughout the central nervous system. And so here we see them in a nice little picture, right? So we have our astrocytes here, look like a little starburst, right? The oligodendrocytes are actually hanging out right next to, kind of connected to the myelin sheath. The ependymal cells over here in that orange, and then the microglial cells, right, even smaller than everybody else involved, right, little guys right there. When we look at the glial cells in the peripheral nervous system, we only have 2. Nice short list. The satellite cells and the Schwann cells. The satellite cells, these are going to surround the cell bodies, give some support. We see these with the sensory and the autonomic ganglia. And then the Schwann cells are kind of the peripheral nervous system's equivalent to the oligodendrocytes, in that they're going to produce that myelin for the axons. So here we see the satellite cells, they're kind of like, just glomming on to that cell body, right, kind of covering it like almost like a little starfish. And then the Schwann cells right there covering and providing that myelin sheath. Now, why myelin? What's the whole point, right, of myelin? The myelin provides insulation for axons, whether it's provided by the oligodendrocytes in the central nervous system or the Schwann cells in the peripheral. But that myelin is a very lipid-rich sheath, so it's a fat-based sheath, and it's going to help facilitate, it's going to help transmit things faster. So the little electrical signals that travel along the axon can travel faster when myelin is involved. And so the nerves that have myelin are called myelinated nerves, right? That glial cells wrapped around the axon. There really is like no cytoplasm between the glial cell layers, and that axon, that segment, is like completely covered by myelin. The unmyelinated nerves, the axons are not completely covered. So there's one side of the axon that is unmyelinated. So keep in mind, there's still some myelin on the unmyelinated nerves, but it's not, there's a section that doesn't have any, right? And we're not talking about the nodes of Ranvier, we're talking about like a side bit. So this is like what we're talking about with the myelination. So we're kind of like, it's almost like wrapping around it, right, if you think of it. So here, right, we see our axon in here, right? We see the axolemma, that's just the plasma membrane of the axon. We see the myelin sheath, right? And it's just kind of like spiraling around it, right? If you were to like coil it around, that's how we're going to end up with myelination. That's how we get the myelin on the axon. But here's that unmyelinated axon. So, like, again, remember I mentioned earlier, unmyelinated, we're not talking about nodes of Ranvier. We're talking about, like, there's a whole side where the axon is exposed. So there's myelin around most of it, but there's a whole longitudinal section of that axon that is not covered by myelin. Now, these axons can experience some regeneration because nerves do get damaged from time to time. And luckily for us, they can repair, they can regenerate to some extent. If it's in the peripheral nervous system, the nerve is damaged, the axons that are distal to the injury, so down the line, right, further down the line from the injury, are going to degenerate, right? The Schwann cells that were around the injured axons, they're going to form a new tube of myelin for growing axons, right, to come through. So a lot of times these axons that are like proximal to the injury can actually cross the injury site and grow back to the original targets. So basically what we're saying here is you get, let's say you injure something in your wrist. And so if you injure a nerve, let's say in your wrist, and it's supposed to continue down your pinky, right? Let's talk about the ulnar nerve. Ulnar nerve runs along the ulna down through your pinky. Let's say we damage the ulnar nerve at the wrist. The axons in the hand and in the pinky will degenerate. But the Schwann cells that were around those particular axons, they're going to then start building up. They're going to form a new myelin tube so that the axons from above your wrist in your forearm can travel through that new myelin tube and hopefully potentially reach through your wrist into your hand, into your pinky, right? So if it was 100% successful, you'd have new axons that could basically move down, right, down into the area that was damaged. With the central nervous system, right, the nerve gets damaged, the oligodendrocytes are going to go through apoptosis or become inactive. So the nerve or the neuron is done, the oligodendrocytes with them are also done. This means scar tissue. So we get a little bit of scar tissue, and then those astrocytes from earlier, they're going to get activated, and they're going to stop the axonal growth. The injured ones are not able to grow back, and the damage is permanent.